A sleep lab produces a polysomnography which records characteristics of the sleeper's sleep throughout the night. It generally has at least 2 lines of readout for the EOG, the electrooculogram. And this measures eye movements, oculo, okay. At least one for the EMG, and that would be the electromyogram, which measures muscle tone of the chin and jaw, and at least one for the EEG, electroencephalogram, which of course measures brain activity or brain waves. So at the very minimum, we've got 4 rows. We've got a left eye EOG, a right eye EOG, EMG, and EEG in that order on the polysomnography. Even more typically, there are 2 rows of EEG, one for the posterior portion of the brain and one for the frontal brain. So a minimum polysomnography would be 4 rows of readout, but most typically they have 6. Here I am just showing you where we typically place electrodes. An EEG may have as few as 4 electrodes, such as this very simple picture I'm showing you of one side of the face, but it can go up to over 250 electrodes. Note that the electrodes for the EOG, the one for the eyes, generally include one in the middle of the forehead too, just for reference. Then the main recording ones can be placed above and below the eyes, or, as shown in this case, to the sides of each eye. Note that the standard EMG electrodes are placed on the chin and the jaw. Now, in a clinical sleep lab, there is an additional goal of identifying sleep disturbances and disorders. Thus, a clinical polysomnography includes additional measures to this end, such as those that measure breathing. You know, there are masks and other devices that measure oxygen intake and breathing patterns. Heart rate and rhythm measured by an EKG, leg movement measured by additional EMG electrodes placed over the legs, etc. Now we're able to send participants home with portable equipment at times, such as masks that collect data over many nights on breathing oxygen levels. Even watch-like devices like Fitbits can measure duration and distribution of sleep. They work adequately for some cases, but in most cases, the accuracy and precision of measurement of the sleep lab is needed. Now, one lab has come out with a home-based cap for which precision and accuracy is a bit better, but to date, no such devices match the precision and accuracy only obtained by a full polysomnography in an on-site sleep lab. So a typical polysomnography shows the sleeper's EEG, EOG, and EMG variations over the entire duration of a sleep period. If I were to show you an entire polysomnography for a sleeper, it would take many, many slides. Here's a snippet of one, and a great question. Can you guess which stage of sleep this snippet represents? This is in fact N3, stage 3, and it is especially apparent looking at the size of the EEG waves. We're going to cover these stages next. And of course, a clinical polysomnography shows the same plus additional measurements needed to diagnose specific disturbances and disorders. Now, a hypnogram is different from a polysomnography. It shows the amount of time a sleeper spends in each stage of sleep during the sleep period and the sequencing of those stages. A typical hypnogram for a healthy adult and for an entire full sleep night looks something like this one on the right. Hypnograms are often provided by apps and wrist devices that you can buy and wear. It is interesting and informative to see the overall quality of your sleep. But as you can see here, the polysomnography as used in an on-site sleep lab provides significantly more detail, information, and accuracy. Now let's look at the different stages of sleep. First, when you are awake, you have very fast and frequent small brain waves, very fast and frequent. But then you get drowsy. And as you progress deeper into N1, N2, and N3 sleep, your brain waves get slower and larger with each stage. Note that at any stage of wake or sleep, your brain shows mixed waves, but often one type is pronounced. When you are awake, beta waves are predominant. They're the fastest, smallest, and most frequent. But when you get drowsy, alpha waves are predominant. So these are a little slower, right? A little larger. The drowsy stage is an awesome state of relaxation, one when you feel like you're just sinking into your bed. Some people learn to meditate in order to hang out in this alpha stage. When you move into N1, you are experiencing what we call light sleep, and theta waves are predominant. Theta, T-H-E-T-A. I could usually easily awaken you in the sleep lab if I were to say something softly into the microphone. Stage 1 is often characterized by something called hypnotic jerks, also known as sleep starts and myotonic jerks. These are brief hallucinations which may be of falling or floating, usually accompanied by a jerk of the body, which may or may not wake you up. N2 is the next stage, and this is the only stage in which we do not have a clear predominant kind of brainwave. Waves are mixed. We see all kinds of them here. This is a deeper stage of sleep than N2, and a little harder to wake you up by just talking softly over the microphone. But I might still, if I say your name, because we are conditioned to react to our names. N2 is probably the easiest to identify with an EEG readout because it is characterized by something called sleep spindles and K complexes. Sleep spindles are tapered at both ends, so they resemble a spindle. K complexes are the largest events in an EEG. Both of them are symmetrical. The functions of sleep spindles and K complexes are not fully known, but research indicates that both are involved in inhibiting cortical activity, allowing us to sleep in one long consolidated period. Sleep spindles often occur following muscle twitching, so it is likely because of sleep spindles that lesser muscle twitches do not normally wake us up. K complexes often follow outside stimuli that do not actually wake us up, like a partner changing positions or the flush of a toilet in a connecting bathroom. So it is likely because of K complexes that we can often sleep through such outside stimuli. Sleep spindles are also involved in learning and K complexes in memory. Learning improves after sleep with more sleep spindles, and memory consolidation improves with more K complexes. We'll be looking at this more closely down the road. Next, you will move into stage N3, and delta waves predominate your EEG. They are the slowest, largest, waves of them all. Now, some books and resources still refer to stages M3 and M4. M3, when delta waves predominate but are less than 50% of your total different waves, and M4 when data waves are 50% and higher. But what was once considered two different stages of deep sleep, scientists now refer to collectively as M3. So M3 is your deepest sleep, and it is considerably more difficult to awaken you in the sleep lab during this stage. REM stands for rapid eye movements. This stage of sleep can be difficult to differentiate from wakefulness and even sometimes N1 on the EEG because your brain is highly active. It actually more closely resembles your brain waves when you are awake than when you are asleep. The best clue to differentiate these is that REM waves are sawtooth shaped. Thus, REM waves are often called sawtooth waves. This stage is characterized by rapid eye movements, muscle twitching, irregular heart rate, and irregular breathing, dreaming, and very low EMG, so low, no discernible reading because of the necessary blockage of final nerve passages from the spinal cord to the muscles. And even though your brain is very active, it's engaged in the inner world, not the outer world, right? REM sleep is often abbreviated as REMS. N1, N2, and N3 are often collectively abbreviated as NREMS, non-REM sleep. Also know that N3, or deep sleep, is often referred to as SWS, which stands for slow wave sleep. In our next segment, we will look at these stages and learn to recognize them in a polysomnography.